First of all, just by way of disclosure, I'm a full-time employee of the United States government, no relevant commercial interests to disclose. Want to talk a little bit about FDA's efforts to facilitate the development of cell and gene therapies, talk about the particular importance of manufacturing, because at least from my perspective, a lot of our issues today end up sitting in that place. Talk about the applicable regulatory frameworks and talk about some resources for product developers. Kind of the bottom line up front here is that FDA is very committed to advancing the development of safe and effective cell and gene therapies for people with unmet medical needs, particularly with those with rare diseases. And we're going to keep doing that by encouraging sponsor interactions with the agency, using all of our applicable development programs, the expedited development programs, and sometimes taking appropriate action when needed to protect patients against unscrupulous actors. Gene therapy has continued to grow. In terms of cell and gene therapy here, you're looking at the investigation of drug applications. You'll see a little bit of a plateau in 2021, but there was something going on in 2020 to 2021 known as pandemic, which did have some effect here. If I would include the expanded access requests during this time in terms of the investigation of new drug applications, this graphic on the left would be significantly higher, and it would look actually like we had a large increase. But trying to be fair here, I've kept those out. But when you look on the right panel at our IND amendments, which are in terms of the way we sort them out, it's hard to sort out what was expanded access amendments and not. You can see here end over end year growth. To wit, we now have 8 approved gene therapies in the United States. Now, 6 of these 8 are cell-based gene therapies, that is chimeric antigen receptor T cell therapies. 1 is a locally administered gene therapy, Luxturna, and 1 is a systemically administered gene therapy, Zolgensma. But this, we're starting to actually have a portfolio of genetically modified cell therapies and gene therapies, which we'd like to try to accelerate the pace of, because of the potential for these products to bring significant clinical benefit. I'm going to start by talking about CAR-T cells and then move on to talk about gene therapies. One of the advantages of genetically modified cell therapies is that whether they are autologous or allogeneic, you can use appropriate methods to figure out where you've made the genomic integration into the cells. You can do this modification outside of the individual. You can potentially make multiple modifications to the cells. You can put switches into the cells to potentially kill them by, if necessary, by administering a small molecule drug. So lots that can be done with genetically modified cell therapies. And in fact, the nice thing about this also is that you can have extended duration of therapeutic benefit, although we're not looking for lifelong benefit. It's just a long enough benefit to, for instance, treat a cancer or perhaps an autoimmune disease. Now, when we move on to challenges here in development of CAR-T cell therapies, some of the things we see is the transition from pilot scales to commercial manufacturing can be challenging. And that's brought up this whole issue of centralized versus distributed manufacturing. There are also some need for novel approaches to clinical development in order to deal with trials, although by and large, when these therapies work, the trials have not had to be very large. People are using closed systems or semi-closed systems to manufacture these. This brings up this debate of centralized versus decentralized manufacturing. Even some centralized manufacturers are using these types of closed systems in a central location just because the need to keep products from individuals separated during the manufacturing process. So just to show you that there is this capacity potentially to manufacture locally for autologous cells, although by and large now we're starting to see these CAR-T cell therapies made remarkably rapidly compared to several years ago, that is within a week or so, at central facilities, and by using all of the logistics to square that away. What is happening is that we do have the hope eventually to see CAR-T cells used for solid tumors. However, there have been several challenges that have hindered the development of CAR-T cells for solid tumors. All of the ones we have on the market today are for hematologic malignancies, primarily non-Hodgkin's lymphomas, acute lymphoid leukemia, and multiple myeloma. There are ways, though, that solid tumors are more difficult to approach that distinguish them. Then that's why we have the lower hanging fruit of the hematologic malignancies. For solid tumors, there's often an immunosuppressive microenvironment around the tumor that is problematic. And so getting good T cell function is a problem. But the biggest problem here really is that solid tumors generally don't have the advantage that some hematologic tumors have of unique cell surface molecules that you can target and essentially leads to killing of those cells. Now, the whole idea of allogeneic CAR-T cells, that's off-the-shelf products that would come from somebody else who's healthy, perhaps, that would be genetically modified to address a tumor, either could be a hematologic malignancy or could be a solid tumor, has become possible in part because with molecular biology, we can manipulate the major histocompatibility complex class 1 molecules on the cell surface and allow one to administer a cell like this that could potentially attack a cancer. The other advantage of allogeneic CAR-T cells, besides allowing you to do some of these, the genetic manipulation, additional genetic manipulation, is that you can have manufacturing consistency. Granted, you can't have just one product because you will have multiple, multiple donations over the course of time. But each lot that you have of that product, I guess it's not just one. It is all just one product, but it's different lots. You can't just have one lot. You can have multiple lots from different donors. That allows you to treat 100 or so, perhaps, patients from one donor. Could be much nicer from the manufacturer consistency standpoint. The nice thing about allogeneic CAR-T cells, they are immediately available for those in need. And they ultimately, the hope would be, could reduce the cost of therapy, which would be nice because if the cost of therapy came down, we might start to see these products used in earlier stage cancers where they actually might work better, particularly on the solid tumor end. The other nice thing about this is that once you have an allogeneic cell line, now you could do this in an autologous setting. It just gets, is the juice worth the squeeze? You have to do a lot of work. But in an allogeneic setting, you can make very complicated targeting constructs to overcome some of the problems that we've had with solid tumors. And what can you do? You can almost have Boolean logic, ors and ands and nots. So you can have CAR-T cells that recognize 2 cell surface receptors in order to cause killing, or you can have them recognize perhaps 3 cell surface receptors. If it sees 2, it knows to kill, but if it sees the 3rd on that same cell, it will not kill. That will be an inhibitory signal. And that kind of logic is what may be necessary to develop these cells to effectively address solid tumors. We still are in the early stages here, and we'll see where this goes. But being able to hit multiple receptors may be what is necessary in order to target solid tumors, which you can't find a single antigen on most that is ideal to target. But maybe if you have 2, 3, or 4, you can find a way around that to a very effective way of getting to killing. Obviously, there's some other challenges to overcome, but this is at least helping to overcome one of the most important of them. I'm going to turn now to directly administered gene therapies, just noting that the whole issue here of CAR-T cells, you can see it's become a somewhat more mature field, and it's progressing. The directly administered gene therapy field has not progressed quite as rapidly, in part because of challenges with clinical development, but in part because of challenges in manufacturing, and I'll tell you more about those. But I want to start with telling you about the success story that to me is what gets me excited and motivated to try to help drive the field forward, because when you can see success like this, you can feel it and can understand what is possible with gene therapy if it works, and why we'd like to see more of it work. So anasemnogene or Zolgensma is a gene therapy that was developed for spinal muscular atrophy type 1. For those who are not familiar, this is actually one of the more common rare genetic disorders, and it affects type 1 of this disease affects infants within the first few months of life. What happens is they don't make developmental milestones, so they don't sit, they don't stand, and they become floppy infants, eventually requiring ventilatory support, and they usually die by about 3 years of age. So very traumatic for the parents, very terrible experience all around. Now, there was an oligonucleotide-based therapy that came up for this that required repeat administration, but a gene therapy was also developed. And the left-hand panel graphic here is using a scoring system that was developed at Children's Hospital Philadelphia to look at the disability caused by spinal muscular atrophy, and an arbitrary scale here, but if you look at the 40, it's not arbitrary, but for you looking at it, it's arbitrary right now. Sorry, but the number 40 here, if you're below, you're abnormal. If you're above, you're normal. You can see that after administration of the gene therapy here, the large majority of children became normal on this scale as they went across time. And to see a child with normal function at 2 to 3 years old is not heard of with SMA type 1. This is not something that has kind of variable penetrance. By definition, these children should have either been on a respirator or been unfortunately close to death or dead. So the picture on the right of a child running around at age 3 is quite remarkable. This to me is what gets me pumped about why we need to think about the potential for gene therapies. And I don't believe this is an exception. I think this could be a rule if we get our act together and overcome some of the challenges that we have. The reason for getting so excited about trying to deal with these rare disorders is, first of all, they're accessible using our current manufacturing technologies in terms of how we could make enough gene therapies if we put our mind to it. But second of all, dealing with these rare diseases actually in aggregate means being able to address literally thousands and thousands to millions of people who have a variety of rare genetic disorders, but it also serves as the basis for how we may have to proceed in the future when we're thinking about using genome editing to deal with more common disorders. Because if you think about it, many more common disorders are a grouping of rare genetic mutations, that is base pair changes, either base pair changes, insertions, or deletions in our genomes, that if you address with a genome editor, end up being a small cluster of disorders together that give one disease, either the different genetic mutations that are causing it. So by figuring out how to address rare disorders, we're hopefully developing the paradigm we need for larger populations too. And really what we're trying to shift from here is this concept of personalized medicine, which is very exciting. Personalized medicine in the 90s, this concept that you'd have drugs on the shelf and you would take them off the shelf based on biomarkers that you identified in people to use. Very exciting. But where we're moving to is this idea of individualized medicine, where we create the right drug to treat the patient. And individualized medicine is like moving from ready to wear into bespoke tailoring or made to measure. And what do I mean by that? The made to measure products are really customized products, which we've had around for some time. These would be personalized cancer vaccines, or perhaps even when you think about autologous chimeric antigen receptor T cells, they're a kind of customized product as well. But the bespoke products are really created products where we're really starting over again. We might be using the same cloth, that is the same vector, but we're going to cut it differently, that is putting in different inserts for different individuals. The challenge, though, of getting there, now you could say the challenge is, gosh, you got to have non-clinical models that help you understand what's going on. The clinical development, oh my goodness, you have to understand the natural history of disease, and you have to be able to look at the changes that you have either in a biomarker that you trust as a surrogate or as a clinical endpoint, and ultimately you have to be able to have product access. All of those things are real challenges. But manufacturing has turned out to be one of the greatest challenges, because if you don't have a high quality product that you trust, you can't figure out whether it's actually working in people when you give it to them. And the manufacturing problem here is that currently, commercially, the sweet spot for manufacturing is the same place as when you go into a certain type of coffee establishment which is ubiquitous. You go into that coffee establishment and you look up on the board and everything is the medium size. They don't even put prices for the large and the small size anymore up on the board because that's the sweet spot. For gene therapy, it's very similar. The sweet spot for manufacturing for commercial viability is currently making couple hundred doses a year. Making tens of thousands of doses is beyond our technical capacity, particularly for systemic administration. And we'll have to work further to get there. But I'll leave that for another day. What I'm more interested in is talking about how we're going to deal with the small size, which is really an issue because there, the commercial viability has not been established. Making these small runs is not something that has been feasible, because every time we start fresh when we make a new gene therapy often, and that means that the setup costs get to be just too high. And when you try to put multiple gene therapies through this process, costs start to become a real consideration. So the problem here is, how do we fix that? We have a problem in terms of getting product to use in patients in investigational setting, and then potentially in commercial setting. So one of the ways that we might think about getting this to move forward is by starting to standardize processes to the extent that you can make a device do some of the things that is necessary. Now, this is a Coke machine or a soda machine that you can program in various ways to get various combinations of drinks. Apparently, it's about something like 1,000 combinations of drinks from this machine. Not that I put this here as a placeholder to say that there are academics and companies working on purification systems that could be automated, which then would allow one, in combination with upstream technologies that we already have, to put together essentially devices that would allow you to produce gene therapies, hopefully more reproducibly, or certainly at least help you handle the downstream processing more reproducibly. That might allow us to start to move through gene therapies one after the other. But that alone won't do it. Some other things will probably also be necessary. We probably need to all start to settle in the academic setting and possibly in the small industry setting on trying to use same methodologies as we develop and manufacture these bespoke therapeutics. This is kind of a pre-competitive area where if we had alignment, the ability to transfer products over to commercial manufacturers, contract manufacturers, or technology transfer to a potential acquirer of a product would potentially benefit everyone because you'd actually see products coming forward more rapidly, and the innovation would shift from spending a lot of time on making the thing to starting to think about how you come up with the best concepts, therapeutic concepts that could address the disease at hand, and the best clinical development plans. The other piece of this is how we and the regulatory world could help with this. And we're trying to find ways that we might be able to allow people to leverage the non-clinical and manufacturing data from one application to another. The concept here is that if you have a very well-described product, that you understand the nature of the vector, you understand what size inserts can fit in that vector, you might be able to start to swap out the insert that's giving the therapeutic effect, and do that multiple times while relying on all of the information you need for manufacturing of the vector from the original product. Could save a fair amount of time and money, and we want to see if this can be validated. Now, the bespoke gene therapy cookbook is this concept of, I bring this up because, you may laugh, but this molecular cloning manual by Maniotis and colleagues, made a really big difference. The reason why it did was because suddenly, when everyone was speaking the same language and using the same techniques, you could share things more easily, and the innovation came into the kinds of experiments that you did, not the solutions that you used to lyse your bacteria, et cetera. Same thing here. The bespoke therapeutic concept of reusing the vector is one, again, to try to push this process so that we spend more time on generating the real innovation, which is the construct with the insert that actually addresses the disease, and less time in essentially doing regulatory paperwork and rework for submissions. This whole kind of concept is being tested in a public-private partnership that's being run out of the Foundation for NIH. They are sponsoring the Bespoke Gene Therapy Consortium, which now has quite a number of both company and nonprofit collaborators working in conjunction with NIH and some FDA input here. The idea here is, can you demonstrate that what I've just told you actually works, that you can actually improve the vector technologies, improve the manufacturing technologies, and then use a standardized process to move things forward. So hopefully that will all get us move forward to a place where we can move things forward more rapidly. And we'll do so within our regulatory framework for biologics. Now, our framework, I just put this up here just to remind you that we do a fair amount of guidance. And where does guidance come from? Guidance is basically something that FDA has that we give to sponsors so that it helps them understand how to stay on the right side of our regulation and rules. Regulations and rules are made by notice and comment rulemaking. They're in for FDA and 21 Code of Federal Regulations. And we have the authority to do that because of the laws and statutes, or laws or statutes, the same thing, that support us, the Public Health Service Act and the Federal Food, Drug, and Cosmetic Act. One of the things that Congress has also given us on top of that are certain, the ability to have certain expedited development programs. The one that I want to highlight today is Regenerative Medicine Advanced Therapy Designation, because that is a program that was started as part of 21st Century Cures in December of 2016, which is specifically tailored to cell and gene therapies that are targeting serious or life-threatening diseases. We try to articulate the nature of this through our regenerative medicine guidance documents. There's a suite of guidance documents that we put out that apply both to cellular therapies and to the genetically modified cell therapies and gene therapies. Our goal here was to clarify our regulations and to help foster the field to move forward. So I'm not going to go through all of these guidances, but just to say that they help, hopefully, to clarify how one goes about developing both cellular products and gene therapies. But the regenerative medicine therapy guidance, which applies to cell therapies that require licensure, therapeutic tissue engineering products. It also applies to genetically modified cell therapies and gene therapies that produce durable effects. That is very similar in many ways to the breakthrough therapy designation, but it's tailored for the cellular and gene therapy products, in part because the clinical evidence that's necessary allows one to look at diseases that may never have been treated before. You don't actually have to show that you're better than a standard of care. You basically just have to show that you're addressing unmet medical needs. You also, with this particular designation, are able to use some expanded methods of fulfilling the criteria for transition from an accelerated approval to a full approval or traditional approval. That was put in place because it was realized that in some cases for these genetic therapies or cell therapies, there might be very small populations treated. And so one way of moving from an accelerated approval to a traditional approval might just be to follow the people you treated in the first place for a longer period of time and just see that the therapeutic benefit remains. And so that's one of the things that can be done. You can see here that at our center over the course of time, since the Regenerative Medicine Advanced Therapy designation was put in place, we now have the number of designations that we've given has exceeded the number of breakthrough designations. That makes sense. The interesting thing is the percentage that we give of breakthrough therapy designations and regenerative medicine and advanced therapy designations is roughly the same. About a third of applications, maybe a little bit more, receive the designation when they come in for either one of these. Here you can see that right now we're running at about 38% of requests granted. That was data as of March 1st, with 68 RMAT designations granted out of 180 requests. We also in this guidance talk about how one can think about doing distributed manufacturing. I'm not going to, for the sake of time, I'm not going to go into this much more, but just to say that there are ways that academic institutions can potentially collaborate and manufacture locally, do clinical trials together, and then each get licenses to produce products relying on a combined data set, something that may be of interest moving forward, particularly for products that require simpler manufacturing. We've also put out some recent guidance on both development of CAR-T cell therapies. These are draft guidances for comments, and we also have the a draft guidance on genome editing products. They're, again, relatively cutting edge. Be interested to see what we get back for comments, and then after we get back comments, we will put out final versions. Finally, I just want to say that in this area, we realize very much that dialogue with the agency is needed, and we realize that one of the great limitations or failings that we've had during the pandemic is our bandwidth to have as many meetings as we really should be having with industry. And that's hopefully something that will be solved by both staffing up and hopefully moving out of the depth of the pandemic. But we have certain types of meetings that people can take advantage of, including our CBER Advanced Technology Team meeting. This is when somebody has a platform and would like to have a discussion about how that platform might fulfill a medical need. And it's actually, it's been very educational for us because sometimes we learn about very interesting manufacturing technologies or gene therapy vectors or other technologies. Very easy meeting to have because we don't require much more than a slide deck and a few questions, and a group of manufacturing experts and sometimes clinical experts are able to have a meeting. This is really, these are always true meetings. These are not written responses or anything. These CAT meetings are usually hour and a half meetings with the agency on these topics. On the other hand, we also have the Interact program, which is our meetings around specific products to give manufacturers feedback or sponsors feedback on anything from non-clinical development to manufacturing or even clinical development plans around a specific product. Here, we require some information to understand what the product is, and we usually get in some questions in advance and then try to answer those questions. Sometimes now we've been doing these mostly by written response because we've been overwhelmed with the pandemic, but we really want to get back to doing these more as live meetings. So really just to summarize, we are very excited about the field of cell and gene therapy. It's got, I think, tremendous potential. We do understand that there are some real challenges to be overcome, but we think that working together, we will see these overcome. And despite the fact that it's been 30 years or more in coming, we really are starting to see gene therapy become something of note in medicine. So thanks very much.
Peter, I wanted to just go over one of your initial comments when you look at the number of therapies that are approved with respect to CAR-Ts. I know you have draft guidance, but should we infer that maybe the agency is pretty comfortable with the technology kind of as is now, or do you look at every single application still sort of starting from kind of square one?
Yeah, no, really, really good question. So I think we look at every application from stem to stern, but that said, because we're so much more comfortable now, doing that inspection takes a lot less time than it did at one point, because now we know where the weak points are to look for. So we are still thorough in our review process, but we're able to do it much more agilely than we were previously. Now, there are some questions that have come up, and you might see guidance in the future about how we might be able, even in that CAR-T cell world, to be able to leverage applications in a manner similar to the way I discussed for gene therapies. Again, trying to allow iterations that bring more innovation without sacrificing safety. So we are constantly thinking about this in terms of how to get regulatory efficiency.
Gotcha. And just on that respect, with regard to safety, that typically is one of the primary reasons why you see a program kind of derailed. And I know I want to get your perspective on the recent, or I guess last fall, the AAV meeting that talked through some of the safeties and kind of theoreticals. Just help us with kind of where you are with respect to that technology and give us some context maybe for that meeting.
Yeah, so great question. AAV, lentiviruses has become the workhorses for transforming, for getting gene therapies into cells. AAV has become the workhorse for in vivo gene therapy, directly administered gene therapy. And there are a lot of programs using it. The question is, why did we have this meeting and we went through all these different potential adverse effects? Do we have problems with the technology? And I think the answer is no, we don't have problems with the technology. I think we're dealing with what any field maturing would have in medicine. We're starting to see AAV applied frequently enough that we're starting to see where its limitations are, how it could be improved, what might work better if it were potentially modified in certain ways. And what are the limitations currently, and where do people need to research to overcome those limitations? So I don't think that anything we saw makes me think of this as a showstopper. I do think it makes me realize that yes, our current AAVs do have some limitations. They do have some things that we have to overcome. And it does make me excited to think about moving to next generation vectors for gene therapies and the continued research into them. But I think we're not. It doesn't put us off at all in terms of the field. And if anything, as we see more results come out from different trials, I think we're excited to see where this field could go. I think it is certainly one where there's a lot of benefit risk that has to go into thinking about things here, because for some cases, although there might be a small risk of something on the downside, there is oftentimes a lot of benefit on the upside.
That makes sense. And I wanted to ask you, there have been a number of, over the past, say, 12 months or so, a number of programs in clinical development that have been paused. And I know some of this may be related to the application itself, not singling out a single drug. And some of it, may be just related to some friction with respect to or inconsistencies getting, the whole pandemic process, everyone's converted to virtual and there's less sort of FaceTime and more sort of, let's say, volatility and kind of, what a natural sort of dialogue would be. So help us with kind of, how do we read into the, the more recent pauses? Is it COVID related, or is it the drug itself, or is it a little bit of both?
Yeah, so I think we have to, I can't speak, I won't speak to any specific programs, but I can say that, because I can't, but I will say that in general, look, we sometimes pause programs when we see findings that we don't understand. I do think that there is a combination of things that has occurred during COVID. In some cases, the natural state of events, something's happened that has been really concerning. We've put something on pause. That's normal until we can figure it out. I do think that COVID has taken a toll on us. Our reviewers have been very strained. We are somewhat short-staffed. And so some of the dialogue that I would have liked to seen that might have helped avoid some of these things might not have occurred. Just have to be completely transparent and honest about that. It's my commitment, and I think the leadership above me is supporting as well, that we will work to staff up and work to support this field in a way that I think needs to be commensurate with what we have in terms of potential. Because we do understand that every time we put a pause on something in gene therapy, there is a collective shudder in the field. We'd like to try to only do it when absolutely necessary to protect health. If we can resolve things in other ways, it's preferable generally.
And just with respect to, the resources to kind of expand the review cycle at FDA, to what degree does PDUFA 7 or funding or other external elements going to help streamline your process?
Yeah, no, we're very grateful because with the PDUFA 7, if enacted and if the commitment letter is accepted through Congress, we would anticipate growing by easily by 50% over where we are staffed now in our gene therapy area. Plus, in addition to that, there are some other headcount that may come there. So we're really considering seeing some pretty significant increases in size of the review team. We're also at the same time working on efficiencies of automation, IT automation that may help us be more efficient. There are certain things that it turns out computers can actually do for us using artificial intelligence that may help free up some reviewer time. The more people we can free up from doing what is essentially clerical checks to be able to be doing intellectually vigorous activities, that would be good. So hopefully all of that's, and by the way, there's support for that IT modernization in the PDUFA commitment letter as well. So that combination is what I'm hoping will allow us to have more bandwidth, much more bandwidth, to be able to engage in the manner that, at least from my opinion, gene therapy deserves.
Perfect. That's helpful. When we look at the gene therapy space over the past 3 to 4 years, there's a dramatic increase in the number of new companies that, preclinical and evolving to clinical stage. Again, not company specific, but have you noticed a trend and any trends really in the applications, what companies do that may be right, what may be wrong? And just given the sort of nascent, where we are in the development of the field, you're naturally going to see a lot of companies that are kind of new to the application process. So maybe what advice would you give some of these startups to help to streamline their process?
Yeah, no, I appreciate that's a really great question, and it's a little bit of painful advice, but the painful advice is actually plan for success for the beginning, which may mean investing some more in the outset. One of the things that we've seen any number of times are people that have developed gene therapies. The gene therapy turns out to work. It seems to work well, in fact. The problem is it's often been produced using a method that is not commercially viable, or for that matter, really acceptable to FDA for use in anything other than academic phase 1 trials. The movement then of that gene therapy to a commercial setting, and sometimes this happens in the commercial setting, where it starts in the commercial setting on small scale and then moves to a larger scale, that can then take years to redo the work, because sometimes it requires then retreating a rare group of patients. And if you're starting with not a lot of patients, having to go back and find more is sometimes challenging. So I think one needs to upfront prepare for success, upfront take the time to have a careful manufacturing process, get the manufacturing process down, understand the potency of the gene therapy that one is using, so that if you do have to transfer process, you actually know where you're starting, so that when you transfer, you know where you end up at, and can adjust that accordingly. But doing that time, setting things up right at the start, rather than just rushing to get clinical results, that time will actually come back to benefit one. It's hard because it's basically it's a human nature to want to get those results fast, but sometimes it's the time you take for preparation is really worth every minute of it. And I kind of use my boating analogy. I've been a sailor for several decades, and when I used to go out sailing, I used to just try to get right out there in the boat, and then would realize, well, my lines weren't exactly right, didn't have the sails on right. This now, before I leave the dock, everything is ship shape, and it makes life a lot easier. It's very similar here in some ways. Just when you leave, depart for the clinical realm, have stuff buttoned down right.
Are there, and when you look at other sectors within healthcare, for example, medical devices, where you have more specific, draft guidance and sort of a more rigorous, kind of process, are there lessons to be learned from other, sectors within healthcare that, we can look to, as kind of a guide for what to expect if a company does meet, in fact, the regulatory guidance?
Yeah, well, I think what we're trying to do is look around us at what's going on in other parts of FDA. There's device regulation, which is different than drug or biologic regulation. I think for what you're seeing here with some of what we're thinking in the way of gene therapies is there a way that we can bring some of the ability to iterate on a given design that we have in the device world to this part of biologics? And some people shudder when they hear that because we generally are biologics and devices are different things, but I do think we want to look closely at whether there are things that we can bring forward here to streamline some of what's going on. Because if we treat every gene therapy as a new entity as it comes through, like a new drug would be, we will never make it through the hundreds to thousands of diseases in a timely manner. So I think we're going to have to find some ways to regulatory streamline, and some of that may be looking at some of how things run in the device world.
And we talked in the beginning, about the leverage, the use of gene and cell therapy, and in orphan diseases, called out a couple of those, and some of the hemonc indications tend to be a little bit more narrow. But as you look forward and say, okay, well, is this technology broadly applicable to much larger diseases? You're not just CNS, but diabetes or metabolic diseases or inflammation, et cetera. What are the challenges in scaling up? And learning the lessons in a more sort of narrow development path to a much broader indication? Is there a path that we can draw from other regulatory, for example, for antibodies, or is there a totally different ballgame in gene and cell therapy?
Well, I think from the clinical development standpoint, there's not a lot in the way here, right? I think it's a matter of how you would end up scaling up the manufacturing and making sure that you felt comfortable with the safety of whatever you used in a larger population. And you'd have to feel like the benefit risk made up for any uncertainty that we have about any issues with AAV when administered to much larger populations, where you might start to see rare side effects that we don't see in the hundreds of thousands of patients who have been treated with gene therapies with AAV to date. But you can imagine there are ways that you could, look, if you had a viral vector or non-viral vector that could deliver a camelid antibody, for instance, that essentially did the same thing that some of our administered monoclonal antibodies do, who knows, bring down certain types of cholesterol. You could imagine that you might have a major paradigm shift there in larger patient populations where one and done, you can fix something. The issue is, how do you manufacture sufficiently with sufficient quality? And then what kinds of, then I think it becomes a question of what kinds of vectors are you comfortable using? Are we comfortable with our current AAV, or do we want to move over to something that may have less potential for concern down the road, including the ability to redose, which is one of the things I didn't spend a lot of time on, but it is one of the limitations of many of the AAV vectors that we're using right now.
That's helpful. Do we have any questions from the audience? There we go. The question is, what are the main reasons why you've declined RMAT requests?
Yeah, so the most common reason is that when we get data in for, if someone brings us data for a relatively uncommon disease, and there's no basis to understand whether the data that they've brought us is changed from what the natural history of disease might have been. So we get very small data sets, just hypothetically, again, not speaking of any one application, but there are any number that come in with, we've treated, 4 people with this gene therapy, and on this biomarker, this looks better. And sometimes, it's better in 3 out of the 5 patients, or, it's very hard to know whether there's a consistent effect. So that's pretty much been the most common reason, is that there's just not enough data for us to feel like there is a consistent effect. On the other hand, we've seen, just so you know, it's not a matter of small numbers, because we have had, people submit RMAT requests with a couple of patients, but because they had baseline data that was very well documented, they gave their therapy, there was a very large effect that was very clearly different. So pre and post, you could tell a very significant difference. Everyone was consistent in the same direction. And, 4 or 5 patients could be fine. So it's not a matter of simply patient number. It's a matter of, sometimes we get results where there's not a major effect, or it's not very consistent.
Thank you. Any other questions from the audience? Just, finally, Peter, when over the course of this meeting, the past yesterday and today, there's been, a lot of, highly innovative sort of permutations of a common theme on gene and cell therapy. Where do you see broadly what are some of the kind of innovations that you think, are they going to add additional risk to the foundation that we already have? Or, do you expect the agency to kind of build upon, I guess back to my first question, build upon some of the commonalities and in technologies?
Yeah, I mean, I'm hoping that as we start to see non-viral vectors, standardized manufacturing technologies come in, that we start to de-risk the portfolio of gene therapies more, and we start to get more comfort here seeing these move forward. Area of tremendous potential here, that I think we need to nurture forward. And obviously we're still early too in this, but we've gotten enough experience under our belt now to understand where some of the pitfalls are. So I think the hope is that as we move forward, we find ways to help sponsors de-risk their development programs maximally. And hopefully, as we advance knowledge more and more, we'll be able to do more and more de-risking.